We left off on Wednesday talking about mechanisms of enzymes. And before we look at one other mechanism, I want to take a little side pathway here and talk about transition state analogs. Um, Basically, transition state analogs are molecules that are made to look like the transition state. And based upon the premise that the enzyme doesn't really bind the substrate, but instead binds and stabilizes the transition state, the general idea is that an enzyme should bind a transition state analog extremely tightly. And in general, that is the case. Usually, there are some actually incredibly effective inhibitors of enzymes that are transition state analogs. Um, an example that has relevance to the medical world would be the beta-lactamase enzymes. And these are enzymes that are often found in bacteria that basically are able to destroy beta-lactam type antibiotics, of which antibiotics like penicillin are an excellent example of a beta-lactam. And beta-lactams are characterized by this four-membered ring that we see right there. And the reactive bond is this bond right here. It's an amide bond, but because normally amides are fairly stable, but because this is in a four-membered ring, and four-membered rings, if you recall from organic chemistry, are typically highly strained ring structures. The only bond in that four-membered ring that can break is that actual amide bond. And as a result, breaking that amide bond is actually part of the mechanism of the beta-lactam antibiotics. Basically a serine present in the active site of the enzyme to which beta-lactam type antibiotics target actually becomes permanently attached to the the carbonyl carbon basically forms a covalent bond, an ester bond with that carbonyl group. Well, in order to do that, you have to have an intact beta-lactam structure get into the active site of the target enzymes. And as I said, bacteria now have the ability to produce beta-lactamase enzymes, which are able to break down the beta-lactam structure, basically hydrolyze it, and thus the beta-lactam antibiotics cannot actually get to their target enzyme. And the mechanism by which they do this is very similar to the enzyme that actually gets inhibited and permanently inactivated by the beta-lactams. And the transition state in the hydrolysis of the beta-lactam is actually relatively similar to what we just saw with chymotrypsin, because we're hydrolyzing an amide bond. And basically what we see is that the serine is going to be attacking the carbonyl carbon here, and you're going to form a tetrahedral intermediate, just like you did, just like we saw in the mechanism for chymotrypsin. So we would expect the transition state to at least have a partial negative charge on this oxygen here. So if we wanted to design a transition state analog, we need to have something that has a negative charge, but also something that mimics the tetrahedral intermediate that we're seeing at this carbon right there. So those are kind of the two things that are needed in this transition state analog. It just so happens that phosphorus forms four bonds with a double bond to oxygen in a fairly stable manner. So molecules like phosphonates, as we see over here, this functional group here, basically we've got the negative charge, which represents the partial negative charge that we see right there. We've got the similar to a tetrahedral structure from having four things bond to that phosphorus. And this molecule bears some resemblance to the beta-lactam, and as a result, if it binds to the beta-lactamase enzyme, it's going to bind relatively tightly. And when it does so, it will usually bind, have a Ki value that is in the nanomolar or lower region, so that you can get incredibly effective inhibition of the beta-lactamase enzyme. And what this would allow is that the beta-lactam would then be able to actually not be cleaved by the beta-lactamase and instead inhibit the enzyme that it's supposed to inhibit. So generally transition state analogs are simply meant to bind extremely tightly due to the fact that they look like the transition state. In fact, more than likely in the active site of the enzyme, there's probably an arginine or lysine residue that is present to stabilize this negative charge that's forming in the transition state. Therefore, the electrostatic interaction between this negatively charged oxygen here and that positively charged group on the enzyme would help this transition state analog bind extremely tightly. So the other mechanism that I want to look at is alcohol dehydrogenase, and this is an enzyme that we've seen before. Basically, it oxidizes alcohols, and this actually illustrates a couple of different things from what we saw with the chymotrypsin mechanism. We'll see the use of a cofactor and a coenzyme. There's a zinc cation present in the active site that plays a role as a Lewis acid, and then NAD plus is a coenzyme in this case. It actually takes part in the oxidation of the alcohol. So if we start off looking at the general elements of the active site, we basically have a histidine residue here that coordinates to the zinc cation. There's two cysteine residues as well that coordinate and hold this zinc in place. And zinc, the zinc cation has 4 coordination sites, and the 4th is taken up by water when the enzyme is not active. In order for the enzyme to become active, it basically has to release this water molecule here, and basically it just gets displaced by the hydroxyl group of the alcohol that's going to be oxidized. So when an alcohol such as ethanol binds, the association between the zinc and this oxygen here cause that alcohol to be deprotonated fairly quickly, and basically the negative charge then is going to be stabilized by both the zinc as well as hydrogen bonding with this serine residue over here. At this point, our NAD plus coenzyme comes in, and it's going to take these electrons along with the hydrogen away from this carbon, and as it does so, it will become reduced, and thus we see the electrons moving around here to give us ultimately NADH. And the next thing that will happen is that in order the carbon would bear a positive charge, but to prevent that from occurring, the electrons from the oxygen are able to move down into form a carbon-oxygen double bond, as we see in the final part of the mechanism here. So over here, this structure would be the NADH, the reduced form of the coenzyme, and here's our product acetaldehyde, which would then be released by the enzyme. So the release of acetaldehyde would be accompanied by the binding of another alcohol molecule, or simply binding of water. So the water or the alcohol will displace the resulting aldehyde.